Hello, I'm Heidi. Thank you so much for joining me today as I share the latest DRAGEN functionality that's coming with DRAGEN version 4.0 and how this new functionality can be used to expand your analysis capabilities across a range of applications. Just as a quick teaser, we have a new suite of pharmacogenomics tools, enhanced single-cell capabilities, including a single-cell ATAC-seq and multiomics pipeline. Our oncology pipeline has accuracy, comprehensiveness, and efficiency improvements. And our PopGen workflow is now faster and more efficient. As you may be already aware, DRAGEN is Illumina's secondary analysis platform that sits between sequencing, so the BCL files coming off the sequencer, then outputting variant calls that can be fed into downstream tertiary analysis and reporting. So DRAGEN offers accurate, comprehensive, and efficient secondary analysis. You can access DRAGEN in a lot of different ways. So I quickly want to touch on this so that we can kind of level set about what we're talking about for today's updates with DRAGEN version 4.0. DRAGEN is available on the latest sequencer of Illumina's NextSeq 1000-2000. And earlier this year, we announced that the DRAGEN licenses are included now with the sequencer. There's no additional cost. So if you have a NextSeq 1000-2000, you have access to pre-configured workflows for a select menu of applications, including WGS germline, some enrichment, germline and somatic workflows, RNA, and single-cell RNA. This DRAGEN onboard NextSeq 1000-2000 offers push-button analysis that's integrated with the sequencer. It doesn't receive every DRAGEN update, though. We aim to update it about once a year. And it has a fixed kind of menu of applications. So for today's webinar, I want to focus on these other access points of DRAGEN: DRAGEN on BaseSpace Sequence Hub, DRAGEN on Illumina Connected Analytics, DRAGEN with the multi-cloud bring your own license on AWS and Microsoft Azure, and then the DRAGEN on-premise server. So these are the ways that you can access DRAGEN that are getting this update with DRAGEN version 4.0. Stay tuned for future updates coming to DRAGEN on NextSeq 1000-2000. Let's talk about how DRAGEN version 4.0 can help unlock the full potential of genomics across a range of applications. In pharmacogenomics, we have an expanded offering that can increase the value of your WGS analysis. We have a new star allele caller for tier 1 genes and a new CYP2B6 targeted caller. For genetic disease, we have increased comprehensiveness of repeat expansion detection, enabling researchers to query 50,000 additional loci for disease correlations. With cell and molecular biology, we've introduced a new workflow that enables the processing of combined single-cell ATAC and single-cell RNA data sets generated from the same set of biological cells. In oncology, we have a lot of improvements and new capabilities. We've got accuracy improvements in HLA typing and tumor-only structural variant calling, efficiency and ease of use improvements in methylation and high coverage somatic analysis, and a set of new fragmentomics biomarkers. For population genomics, we have our joint genotyping pipeline is now faster and more efficient. We won't talk about that in detail today, but there's more details that you'll be able to see in the slides. There's a number of improvements with DRAGEN that don't exactly map onto one application area, and we'll touch on those as well. Although this is a software update webinar, we did recently release our latest DRAGEN hardware, the Illumina DRAGEN Server v4. So we'll talk about that a little bit. We've introduced machine learning in a previous release of DRAGEN, and now it's more efficient and enabled by default. That's an important update that we'll cover in detail as well. We have a new imputation pipeline for human references that enables low-pass whole genome sequencing. Our structural variant caller has runtime and accuracy improvements. We also have introduced a toolkit to allow you to build your own multigenome or graph reference. And our BCL conversion and ORA compression have some improvements where you can now more seamlessly generate ORA compressed FASTQ files within BCL Convert. All right, so let's talk about that new DRAGEN server that we released in June of 2022. It's the latest version of the DRAGEN server. Similar to the V3 server, it's purpose-built to provide accurate, comprehensive, and efficient secondary analysis. It's kind of an incremental update to the existing DRAGEN V3 server with moderately faster processing times. This is entirely because of improved CPU specs. The FPGAs, the kind of hardware that runs the hardware accelerated algorithms, are the same between the V3 and V4 server. The V4 server has some improved data center features, including easier rack mounting, improved BMC, and a dual power supply. And it's compatible with all DRAGEN software versions, similar to the V3 server. So both servers go back to previous versions of DRAGEN and are compatible. So I'll briefly touch on the comparison between the V3 and V4 servers. As you can see, the V4 server has some improved CPU and memory. Its scratch drive is twice as big. It does have the same FPGA card, and both come with the same operating system, CentOS 7. We will be migrating to Oracle 8 over the next 2 years as CentOS 7 is reaching end of life, and both servers will have an upgrade path to Oracle 8. Finally, on the bottom, you can see that the runtime is a little bit faster with the V4 server, which I'll show you a bit more details in the next slide. So when we take the hardware and software updates together, essentially DRAGEN is just as fast, but even more comprehensive with the 4.0 software update. So first, let's look at the comparison between the V3 server and the V4 server. So in light orange compared to dark orange, you can see that the runtimes are a little bit faster, where with DRAGEN version 4.0, it took about 34 minutes to comprehensively variant call a 34x human genome with DRAGEN version 3.10 on a V3 server compared to 30 minutes with the V4 server. So it's a little bit faster. And now we're going to compare DRAGEN version 3.10 software to DRAGEN version 4.0 software. So you can see in both cases, it's about 30 minutes on the V4 server. However, with 3.10, the callers that were enabled for this comparison were small variant calls, structural variants, copy number variants, repeat expansions, and CYP2D6. Whereas for DRAGEN version 4.0 software, we've enabled additional callers, including CYP2B6, GBA, SMN, and star allele. So the runtime is the same, but DRAGEN is even more comprehensive. Okay, let's talk about machine learning. We introduced this as a feature in 3.10, a sort of a beta feature, but we've made some improvements into the efficiency, and now we're able to have that turned on by default for human germline small variant calling. So the plot on the left is something that you might recognize from our version 3.10 software release. This is showing the accuracy, the F1 score of DRAGEN for small variant calling in all benchmark regions from the Precision FDA Truth Challenge V2 compared to the other entries in this challenge. And when we introduced machine learning in DRAGEN version 3.10, we were able to match the accuracy of PacBio sequencing data with Google Deep Variant. So we still have that same accuracy. So we're kind of redefining, setting new standards for accuracy. But now that machine learning is turned on by default. So I'll direct your attention to the right where we're looking at the runtime. So with DRAGEN version 3.10, it took about 30 minutes to map, align, and small variant call with machine learning enabled. And with DRAGEN version 4.0, we've improved that. It takes about 22 minutes for that. And so with this improved efficiency, we're able to turn this feature on by default. There's some important things to note about this change. So the machine learning model is now packaged as part of DRAGEN. So it'll be part of that installer that you get with the DRAGEN version 4.0 software. We now support HG19, HS37D5, in addition to HG38. The germline WGS and enrichment workflows are supported, and machine learning will automatically be disabled for unsupported workflows. A very important note here is that the non-machine learning DRAGEN variant calling scores will no longer be emitted as part of the machine learning workflow. That means that the DGT, DGQ, and DQual for the non-machine learning variant caller are not included in the VCF when machine learning is enabled. So you'll see different quality scores than you would have seen with the 3.10. You can turn off the machine learning with a simple flag. We do see that machine learning improves the accuracy, so we recommend you keep it turned on. And it improves the accuracy for both graph or multi-genome references and non-graph references. So here, I know there's a lot of data shown here, but we're looking at SNPs in the left and indels on the right, and we're plotting false positives and false negatives as measured against a truth set, the NIST 4.2.1, not to be confused with the 4.0 software version of DRAGEN. And you can see across a range of samples, HG001 to HG007, that enabling machine learning reduces the number of false positives and false negatives for both non-graph references as well as graph or multi-genome references. Let's take a look at how this machine learning fits into the workflow. So you can input your FASTQ and reference into MapAlign, or your BAM and your reference into MapAlign, or your BAM and reference into directly into the variant calling. And then the machine learning is kind of an additional step that's now integrated with the DRAGEN variant calling. It takes in the pre-trained model and support files that are part of the installer and runs the machine learning as an integrated step within DRAGEN variant calling. As an output, you get that recalibrated VCF that again has the updated qual, GT, and GQ fields, and should give you improved accuracy of variant calls. All right, let's talk about the updates that we have for DRAGEN across different applications. So DRAGEN for pharmacogenomics. As you know, pharmacogenomics is the study of how variations in the human genome dictate a person's response to medications. In one study, more than 99% of people assessed had a genotype associated with a higher risk to at least one medication. So this impacts a lot of people. And findings from pharmacogenomics research can lead to better future outcomes for both individuals and healthcare providers through improved medication safety and efficacy and lowered medical costs. So we're basically saying that now with DRAGEN version 4.0, you can expand the value of your existing whole genome sequencing analysis by turning on just a few simple features. You can get more information out of your whole genome sequencing. You can genotype difficult but important pharmacogenomic genes with CYP2D6, which already was part of DRAGEN, and CYP2B6, a new with DRAGEN version 4.0, these targeted colors. And then you can determine the optimal genotype and corresponding metabolism status associated with the genotype for so-called tier 1 PGx genes. And this is new with DRAGEN version 4.0. So let's take a look at the DRAGEN PGx offering with DRAGEN version 4.0. There's a few different components to this. First, there's a star allele caller, which calls star alleles and metabolism status for 16 genes. So you can see those 16 genes listed. These all can be called with the same star allele caller, and they provide an output that we'll talk about in some subsequent slides. Then there's some more challenging genes that can't be directly addressed with this single star allele caller. And so we have targeted callers for CYP2D6 and CYP2B6, which call star alleles and metabolism status for these difficult genes. And they output the output as separate output files. Finally, we have a targeted caller for HLA, which outputs the genotype. It doesn't yet output the star allele or metabolizer status. That's something we'll be working on for a future update. So in summary, we have provide the genotype and the genotype for 18 of the CPIC level A genes. We provide the genotype for 20 of the CPIC level A genes, and then the metabolism status and star alleles for 18 of these CPIC level A genes. All right, so the star allele caller, let's dive into that. As you know, star alleles are haplotype patterns of a gene and can correlate with drug metabolism status for genes that are involved in drug metabolism. So the DRAGEN star allele caller identifies the genotypes of those 16 PGx genes that were listed in the previous slide and listed here as well. For each gene, the caller returns the optimal genotype along with the corresponding metabolism status associated with that genotype. And it can be enabled with a simple flag. Today we only support the HG38 reference. It's important to note. So looking at the workflow, we take in genomic data in a lot of different formats. So either FASTQ, BAM, GVCF, or VCF. And the Pharm-GKB star allele definitions are also taken in as an input as part of the config file. The star allele genotyping is performed, and then that genotype is mapped to a PharmCat genotype metabolism status. And ultimately, we end up with the metabolism status and the genotype, the PharmCat metabolism status, as well as a number of other metrics. You can see that across different sample types, we have high concordance to existing methods. Here where we don't have 100% concordance, that 94%, it is because the truth set was from an outdated star allele definition that's been changed. So really we have high concordance to existing methods. All right, let's talk about the CYP2B6 targeted caller. So the gene CYP2B6 is a member of the cytochrome P450 family of important pharmacogenes, playing an important role in the metabolism of a number of drugs of interest, particularly CNS acting drugs with neurologic side effects. Detecting variants in CYP2B6 is complicated because there's sequence homology with the pseudogene paralog CYP2B7. By differentiating between reads mapping to the CYP2B6 gene and its paralog CYP2B7, the new DRAGEN CYP2B6 caller can detect 37 different star alleles known in the population. So it can be turned on with this simple flag, enable CYP2B6 true. It supports the GRCH37, HG19, and HG38. It does require WGS data similar to the other DRAGEN targeted callers. And it's available across the different deployments of DRAGEN. Here again, you can see that across different sample types, we have high concordance to existing methods, including 100% concordance to Stargazer on 7D Coriell samples. So let's just take a look at the DRAGEN PGx workflow kind of in aggregate. You can see we have our regular kind of primary DRAGEN workflow where we input FASTQ data, do the DRAGEN read mapping, and then end up with a BAM file. So this BAM data is then what gets fed into the targeted callers. Because it kind of has to map reads to the gene or pseudogene, it needs that aligned data. It can't go straight from the variant call data. And so the DRAGEN targeted callers, CYP2B6, CYP2D6, and HLA all run on the aligned data, the aligned reads. And then on the variant calling side, so we can perform variant calling, generate GVCF data that then gets fed into the DRAGEN star allele caller, which outputs the genotype calls for star allele caller by taking in the star allele definitions from Pharm-GKB and Pharm-Var. This entire workflow can be enabled with these two flags, enable PGx true and enable HLA true. In the future, we're going to work to having everything enabled with just a single flag and have a unified output for all of the different callers. All right, let's talk about DRAGEN updates for genetic disease. Here, the focus is really going to be on repeat expansions. As you know, short tandem repeats or STR expansions are a major cause of over 20 severe neurological disorders, including ALS, Friedreich's ataxia, and Huntington's disease. With DRAGEN version 4.0, we're introducing Expansion Hunter version 5 that expands the default catalog of genes detected over twofold and supports around 50,000 additional loci. So this support for the expanded catalog is really enabled because of some runtime and memory usage improvements so that we can now look at a much larger STR catalog. The default catalog increases from about 30 to 60 loci. I'll show some details about that on the next slide. And then optionally, users can call repeats on this expanded catalog with 50,000 polymorphic STR loci with minor runtime impact. So it takes about 8 to 10 minutes of additional runtime on by compared to running the default catalog. So here's the default catalog. We have the genes detected in DRAGEN version 3.10 shown in gray, and the new genes detected with DRAGEN version 4.0. So we've included a number of genes of interest, such as BEAN1, a challenging gene implicated in spinocerebellar ataxia. Moving on to DRAGEN updates for cell and molecular biology. We have a new pipeline with single-cell ATAC-seq, which enables single-cell resolution of chromatin accessibility. DRAGEN single-cell ATAC can be run now in combination with DRAGEN single-cell RNA, enabling simultaneous profiling of the transcriptome and epigenome. So as input, the ATAC takes in FASTQ files with reads as triplets, as well as RNA input, and then input reads are specified in a FASTQ list file. The output is a joint count matrix in sparse format for each of the cell barcode that for each cell barcode contains information about how many UMIs corresponded to each gene and how many ATAC fragments intersect each peak. So to turn on this multiomics pipeline, you use the enable RNA flag, the enable single cell RNA flag, and then the enable single cell ATAC flag. You can also enable the ATAC pipeline on its own by just using the enable single cell ATAC flag. This is available on BaseSpace and the multi-cloud bring your own license, as well as the on-premise server. I want to note that the BaseSpace app currently only will support the single cell ATAC workflow, not the multiomics workflow yet. All right, so let's look at the workflow just kind of in a diagram. As mentioned, so the RNA workflow, the RNA single cell takes in RNA, FASTQ reads, performs RNA read alignment, RNA barcode error correction, does the RNA read counting, which then feeds into the cell calling. On the ATAC side, you do your DNA read alignment, your ATAC barcode error correction, and then ATAC read counting, which then also feeds into the cell calling. And finally, the output is a multiomics matrix that includes the cell by gene expression, as well as the chromatin accessibility information. You get your multiomics metrics and single cell ATAC fragments. If you run the ATAC pipeline by itself and don't enable the RNA reads, you still get the multiomics matrix. It just doesn't contain any gene expression information. Okay, let's talk about the DRAGEN updates for oncology. We have some HLA typing accuracy improvements that I'll go into in a bit of detail. We have 3 new biomarkers that quantify genome-wide fragment profile or fragmentomics that we'll go into in detail. And then there's a couple of additional updates that we won't have a chance to go into in too much detail, but you can check out the additional slides on your own time. But wanted to note those here. So we have improved capabilities of doing high coverage somatic analysis. So previously we could only go up to about 600x coverage of whole genome sequencing, but now we can go up to about 1000x. So, enabling detection of even lower frequency alleles. So this is an exciting update for somatic whole genome analysis. Then for the tumor-only structural variant caller, we've got a couple of improvements. So we have a tumor-only scoring model. The DRAGEN structural variant caller tumor-only pipeline previously reported all putative variants in the final VCF file, which could lead to low precision. DRAGEN version 4.0 introduces a tumor-only scoring model, allowing the events to be filtered by a quality score in the final VCF. So essentially, this scoring model has been made consistent with the small variant caller approach and can help lead to better precision in detecting those tumor-only structural variants. We've also introduced a tumor-only systematic noise filtering. So without a matched normal, the tumor-only workflow has challenges removing background noise from the analysis. We now have a tumor-only systematic noise filter that captures mapping and variant calling artifacts and recurrent structural variants, removing likely false positives. And so this is available for download on the support site for HG38 reference and can be enabled with a simple command. All right, so HLA genotyping is notoriously difficult due to high levels of sequence homology between HLA genes and pseudogenes, as well as dense variability. DRAGEN version 4.0 includes improved HLA typing accuracy. So we've introduced a new nucleotide-based alignment and expectation maximization for genotyping, which leads to over 99% accuracy for WGS and whole exome sequencing, and about 97% accuracy on TSO 500 panels. So to go into a bit more detail on this, formerly the HLA genotyping within DRAGEN relied on amino acid alignment with maximal exact match and integer linear programming. With this new HLA typer, it performs nucleotide-based alignment and expectation maximization for genotyping. So this corrects miscalls that were due to incorrect translation. And it's basically implemented the same as the previous component. So it's enabled, you can turn on the flags in the same way. No additional command line options are required. So if you're using the HLA typer, you can now continue to use it with improved accuracy. Here's a view of the performance across a lot of different sample types. So you can see for whole genome sequencing, both for germline, for tumor normal pairs, and for tumor only, the accuracy is at above 98%. For whole exome, both for tumor only and tumor normal pairs, the accuracy is close to 98% or greater. And then for TSO 500 solid and liquid, we're at around 97% accuracy. I want to note that this HLA typer is not released as part of the DRAGEN TSO 500 analysis software, but you could use this tool on your, your data from TSO 500 and get the HLA typing, highly accurate HLA typing results. Okay, so fragmentomics is the study of fragmentation patterns of cell-free DNA or circulating tumor DNA. Fragmentation features are distinct between cancer and non-cancer cells, and studying this can be a powerful tool to infer cancer status and tissue of origin. So cell-free DNA contains molecular features characteristic of their tissue of origin, and different tissues, including cancer cells, contribute cell-free DNA to the circulation. The DNA molecule size, fragment end locations, and end motifs can be influenced by the nucleosome organization, chromatin structure, nuclease content, and gene expression of the tissue of origin. So there's a lot of interesting things to uncover by studying these profiles. And there have been 3 parameters published in the literature that can be measured to quantify these characteristics. So we've implemented those within DRAGEN, and I'll kind of provide an overview of those different metrics. So first, we have the end motif frequency, which is calculated as the frequency of the K-mer motif at the 5' end of the fragments, where K can be specified by the user. Next, we have the window protection score, which is defined for each of a user-provided list of regions. You count the fragments that fully span the region and that end within the region, and then take the difference of the two. Finally, there's a genome-wide fragmentation profile, which is the ratio of short to long fragments in 5 megabase continuous bins of the genome. And so taken together, these parameters can help correlate differences between cell type of origin, cancer type, et cetera. So the DRAGEN Fragmentomics implementation is about 20 times faster than the existing pipelines and has high concordance to the existing Delphi methods. So for example, the fragment profile takes about 19 minutes running with other tools and takes about 30 seconds to run with DRAGEN on whole exome ctDNA data downsampled to 10 million reads. All right, for methylation, we have some improvements as well. We have an improved, easier to use, more efficient end-to-end dedup pipeline. I'll go into some detail on that. And we've also introduced UMI support for methylation assays, which enables more accurate methylation calling by removing PCR error and sequencing error. So the methylation end-to-end dedup workflow is now 1.5 times faster and reduced overhead. So previously, you had to run 2 different steps to get sorted dedup cytosine report. So first, you had to run the alignment from FASTQ to BAM, and then the second run was running the methyl call. And together, that took about 98 minutes to run. Now you can do it in a single step. So it's only charging your license once, and it takes only about 65 minutes to run. So you can see on the right, those commands to enable this workflow, with the key one being enable duplicate marking true. We also now support UMIs for methylation. As mentioned, it removes PCR error and sequencing error. And it has the same principle and requirement as the DNA UMI. So similar designs there. And here you can see the example DRAGEN command line options to be enabled. Okay, let's dive into some of the DRAGEN updates that span applications. Imputation enables accurate genotyping of low-pass sequencing data, making whole genome sequencing more accessible. We've implemented the widely popular GLIMPS pipeline within DRAGEN, and it's about 2.3 times faster within DRAGEN. So let's go into some of the details on that. So we offer an end-to-end DRAGEN imputation tool that's software accelerated. And its key benefits are that it's an end-to-end pipeline with a user-friendly tool to execute scalable whole genome imputation. You can impute a single sample or supply a list of VCF files in a text file format to impute up to 100 samples simultaneously. And so if you run in multi-sample mode, the multi-sample variant call format file is provided as an input to the software. And then the imputation tool imputes the samples one at a time, but incorporates other samples in the multi-sample VCF into the reference panel, using these samples as additional reference data points to aid in haplotype block definition and matching. So you do end up getting more accurate results if you are kind of doing the multi-sample mode. We have a BaseSpace app that's available for running this imputation workflow. And as mentioned, it's about 2 times faster than the original GLIMPS code. So if you're running the workflow on BaseSpace, you can take in your sample FASTQs as well as the Illumina reference panel, and then perform DRAGEN aligning and variant calling, which then gets fed into the imputation workflow. Or if you're running the workflow with the local DRAGEN server, you can take in directly the list of VCFs in a text file format. The imputation workflow then consists of 3 steps. There's a chunk step that partitions the genome into chunks, a phasing step that performs imputation on a per chunk basis, and a ligate step that concatenates the imputed chunks together. And so DRAGEN performs all of these as one integrated step. There's a trade-off between the chunk size and, the larger the chunk size, the greater the accuracy you'll get, but also the longer it will take to run. And so we've done some work to kind of optimize what we think leads to the best balance and trade-off between accuracy and runtime. And so that's kind of provided by default as part of the imputation pipeline. One important thing I want to note here is that the Illumina reference panel is human-based. So we currently only support human samples with this tool. We do know that there will be a lot of interest of this tool within the agricultural space and non-human references. So we'd love to hear your feedback and input on that, of which species you'd most like to see for this. But at launch, we will only be supporting human reference panels. So let's take a look on how this imputation tool impacts accuracy. So essentially, it improves sensitivity of 1x data by more than 70% and reduces false positives by more than 80%. So really what we're kind of comparing to in gold standard in this case is 30x data that's not been imputed. And what we want to see is to approach the accuracy of 30x non-imputed data. So the ROC curve on the right, I'd like to draw your attention there first. And you can see plotted in orange the accuracy of the 1x non-imputed data. You can see it has pretty poor sensitivity, doesn't even approach 50% across a range of thresholds. But when we introduce the imputation tool, we now in blue have accuracy that's much more closely approaching that of the 30x non-imputed. And so this can be a great tool to beef up the accuracy of your low pass whole genome sequencing data for human reference. This software accelerated code also reduces the computational time by about 2.3x. So for multi-sample processing runtime in minutes, it takes about 140 minutes of this data set with GLIMPS versus about 60 minutes with DRAGEN version 4.0. And then to just give you kind of an idea of how long you might expect this to take for different sample types or number of samples, on BaseSpace, the end-to-end runtime for 50 samples is about 8 hours and 45 minutes. If you are going from VCF input, so running imputation only, Then if you're running 100 samples, it takes about 15 hours and 46 minutes. So scaling approximately linearly with the sample time and runtime. And then for 100 samples, if you're running from FASTQ file as input, takes 21 hours and 34 minutes. And then to just give the local end-to-end runtime, so running it on a DRAGEN server, this is on some of our older hardware, the V2 server, takes about 8 hours and 42 minutes to run 50 samples on that server. So similar to BaseSpace for these lower sample numbers. Okay, so we're introducing a new feature with DRAGEN version 4.0 that's a multi-genome reference builder. So the DRAGEN Graph, which is essentially a multi-genome reference. So we've been calling it DRAGEN Graph, but really it's a multi-genome reference. Contains population SNPs and alternate haplotypes to enable more accurate read mapping. So this is something we introduced with DRAGEN version 3.7. We've continued to iterate on this. And now with DRAGEN version 4.0, users can build their own multi-genome reference, which essentially would enable you to diversify your reference genome. So previously, the DRAGEN Graph or multi-genome hash tables were only available via download from the Illumina website. So we created our own multi-genome reference for our customers to use, but now users can build your own custom multi-genome hash table, only for human genomes so far. So HS37D5, HG19, and HG38 reference genomes. Alternative haplotypes and variants for the graph are given in user-provided phased VCF files. So you do kind of have to know how to construct these phased VCF files. So I would kind of bucket this as kind of an advanced feature to be able to build your own multi-genome reference. It takes about 10 minutes for 16 input phased VCFs, for the HG38 V2 graph, for example. So this reference builder that allows you to build custom human multi-genome hash tables takes a phased VCF as input, a graph bed that specifies the regions where alt variants will be included, a reference FASTA that has the human reference genome that supports the HS37D5, HG19, or HG38, and then the mask bed that specifies regions to mask with ends, essentially the false duplicates. So really it's this mask bed, the graph bed, the phased VCFs. There still is some level of expertise required to be able to configure this multi-genome hash table. But essentially it takes in these inputs. The multi-genome file generator then generates the pop alt contigs, the alt contig liftover, isolated population SNPs, and then builds a hash table out of that, which then gives you your custom multi-genome hash table. All right, we also have some updates on DRAGEN ORA Compression. So just to remind you, DRAGEN ORA Compression reduces FASTQ file sizes up to 5-fold. So ORA stands for Original Read Archive. With DRAGEN version 4.0, users can now generate FASTQ.ORA files as part of BCL conversion. So that's something that we already had in version 3.10 as a beta feature, but now it's kind of out of beta. It's a fully fledged feature. It can be enabled in a single command. And so there's some additional processing options. You can specify the number of threads used per file, the number of files processed in parallel. And here's some of the example DRAGEN command line that you would use to enable this. We've also had some requests from customers about implementing or enabling an MD5 checksum of FASTQ.ORA as part of compression. So we've taken that feedback. We've heard that this is important to be able to adopt DRAGEN ORA compression. And so we've now enabled that as part of DRAGEN version 4.0. And so you can use it with this flag or enable MD5 true, and it generates a new file under the output directory that has the MD5 sum. And it has a very modest runtime. It adds about 0.25% to the total compression CPU runtime. An important thing to note here is that this can only be enabled if you're going straight from FASTQ.gz to FASTQ.ORA. You cannot enable it when you're compressing directly from BCL files. Okay, so mitochondrial variant calling. Mitochondrial DNA is of interest for variant calling because mitochondrial disorders are heterogeneous diseases that can arise at any stage of life. Studying mutations associated with mitochondrial disorders can be challenging due to phenotypic variability and genetic heterogeneity among individuals. DRAGEN version 4.0 includes improvements to the mitochondrial variant calling pipeline. So here we're looking at the variant calling false positives and false negatives for both high allele frequency and low allele frequency data sets. And you can see that DRAGEN version 4.0 improves SNP variant calling accuracy in mitochondrial only DNA compared to DRAGEN version 3.10 for both high and low allele frequencies. So I'm looking at DRAGEN version 4.0 boxed in orange compared to DRAGEN version 3.10. And another important note here is that DRAGEN is more accurate than GATK and Strelka. That's true for DRAGEN 3.10 and DRAGEN version 4.0. DRAGEN 4.0 also improves SNP and indel variant calling accuracy in whole genome sequencing data compared to DRAGEN version 3.10 for both SNP and indel. So last slide, we were looking at mitochondrial only DNA. Now we're looking at WGS data. And here we see that we also have a reduction in the number of false positives and false negatives. All right, I now want to just provide an update to some of the new apps that are coming to BaseSpace for DRAGEN. So some of these are specific to the DRAGEN version 4.0 release, but a lot of these are kind of independent of DRAGEN version 4.0 and just wanted to make you aware of some of these changes and new apps that are available. So first we have DRAGEN Germline for PopGen v3.7.8. So this is essentially the germline analysis for whole genome sequencing that's just built off of DRAGEN version 3.7.8. And this is a version that some large scale population genomics initiatives like the All of Us program have been using. And so we wanted to make an easy to use, easy to consume app that has basically all of those same features, same colors enabled, the exact same settings as some of these larger scale population genomics initiatives. So if you want to analyze your data in a manner that's identical to that's used by some of these larger population genomics, you can use this app for that purpose. But it's essentially a tagged specific version of the regular DRAGEN Germline BaseSpace app. We then have another app that's DRAGEN PopGen v3.10.5. So this one is actually running the GVCF genotyper joint calling pipeline. And it has enhanced capability of processing large number of samples in parallel. It can support up to 800 whole genome sequencing GVCFs. So we have these two apps that both have PopGen in the name, but just to clearly distinguish, the DRAGEN Germline for PopGen is running the DRAGEN Germline workflow, whereas the DRAGEN PopGen is actually running the GVCF genotyper workflow. We then have a couple of apps that are supporting the new features and functionality that's coming with DRAGEN version 4.0. So we have our DRAGEN Imputation pipeline. This is new, very exciting. It's based off of DRAGEN version 4.0.3. It has this software accelerated glimpse that we talked about. It can take both VCF and FASTQ as input and has its built-in IRPV, the Illumina reference panel, as well as custom reference panels. So here's where if you had kind of a custom reference that you kind of knew you wanted to use, you could upload your own custom reference panel. And then we have our DRAGEN single cell ATAC pipeline or BaseSpace app that's based off of the DRAGEN single cell ATAC pipeline, new with DRAGEN version 4.0.3. It can take in random or non-random cell barcodes, does peak calling or known peaks and motif scanning in the peaks. So important to note that this DRAGEN single cell ATAC does not yet have the multiomics capability, so you can't integrate with the RNA data. This is specifically for the ATAC data. All right, lastly, we have another BaseSpace app called DRAGEN Germline All Callers v3.9.5. So this one is based off of DRAGEN version 3.9.5. And it essentially has all of the germline callers enabled by default. So on a regular BaseSpace app run for DRAGEN germline, there might be a number of callers that you don't know are actually available to you. It's kind of hard to find them in the settings. And so this is an app that will turn on all of the callers that are there for you. So we have repeat expansions, SMA caller, GBA caller, CYP2D6, CNV, SV, gives you the full range of output of what DRAGEN can give you. And it provides also an integrated DRAGEN report. So it shows these outputs in kind of a nice tabular view. This app can be run straight from FASTQ files. So you can run from input FASTQ files and do all of the variant calling as part of the workflow, or it can accept existing DRAGEN app results if you just want to generate this kind of integrated report of all of the features. And so take a look at that one on for DRAGEN germline all callers v395. We'd love to hear your input or feedback on that. So that's all for the new features with DRAGEN. There's a lot more in the slides that we didn't get the chance to cover today. So if you want to take a look in more detail, please take a look at the slides. Thank you so much for taking the time to join me today. I hope this session has been helpful for you. Thank you.